So first of all, I just want to, it's great to be back here another year, give an update from, this kind of long journey of trying to develop a gene therapy approach for this. It's been a windy road, and this is really focused on just some updates that we've had recently. All right, so these are my disclosures. And, I'll start off because I guess I'm the first talk that's really going into AAV and gene therapy. So just as a little bit of an introduction, this is using adeno-associated virus as a vector. This has become a pretty ubiquitous vector in terms of being used in gene therapy due to kind of some natural features of this virus where it's naturally non-pathogenic, although very common in humans. And at this point, we've got a broad toolkit of different capsids that are available to deliver to different targets. And I'm going to talk about AAV9, which has kind of become the gold standard for delivering genes to the nervous system. There have been over 300 clinical trials initiated with AAV. Overall, it's had a pretty favorable safety record, although there have been some in recent years as doses have been pushed very high in some disease populations that there have been some patients that died in AAV trials. But it also has, starting to gain kind of a regulatory track record where there's now several AAV gene therapy products that have been fully approved in the US and Europe and other countries. All right, and I'm going to talk about kind of a platform approach. Our work with SLC6A1 didn't happen in a vacuum. We've had a history of developing gene therapy approaches for different neurological disorders, and the idea is really just, swapping out the gene, but otherwise using very similar approaches. SLC6A1, becomes a little bit more complicated. Most of these have been neurodegenerative diseases, but SLC6A1, of course, is neurodevelopmental. It is similar to what we're doing with Rett syndrome, which my lab has worked on since 2007. But a number of these have moved into clinical trials, so there's clinical experience of how to translate these into patients. And so everything that we're doing with SLC6A1 here is really benefiting from the knowledge of the work on all of these diseases that kind of came before it. And I put these as, these ones are in clinical trials or have approved for clinical trials. These ones are IND enabling, which means we've gotten very good preclinical data. We're actively trying to move them towards clinical trials. And then these are some earlier stage with discovery. And so SLC6A1 is here. And at this point, we are looking at our steps to try to move this towards a clinical trial. And I'll share the data that supports that here. And I'm not going to take a deep dive. I kind of did a deep dive last year at the symposium, so this is more of an update. But I'm happy to announce that a week ago, basically all of our studies in the mice to date have published as a preprint in JCI. And so this is, and we paid for the open access, so everybody would be able to download this and look at it for free. Let's see. But just kind of a summary of that, our design for this was using an AAV9 capsid, and we packaged the human SLC6A1 gene as a self-complementary genome. So this is a smaller sort of double-stranded genome using the MECP2 or a MECP2 promoter fragment to drive mostly neuronal expression. We had tested another design, but this was the favored one in the end. And just focusing on this, these are some highlights kind of from the paper, and then I'll go into a little bit of new data, is where we were looking at the EEG phenotype in these mice, and we were testing this in knockout mice as well as heterozygous mice. These were the knockout allele, but mice that are heterozygous or homozygous for that. So first of all, if we treated newborn mice by neonatal administration in the heterozygous mice, then we went from this very severe, this is the example EEG tracing from mice injected with vehicle, and then this is with the vector, and we quantified this here where we saw, again, dosing newborn mice, basically complete amelioration of the EEG, abnormal EEG phenotype. If we treated homozygous knockout mice, now these are much more severe model, more severe than the patients, where, if we record, these spike trains, about 50% of the time these mice have these spike trains. Like it's very severe in these mice. And when we did a neonatal ICV injection, then we were able to get a significant reduction in that in that EEG pattern. All right, and then we went through, I'm not going to go through all this, but we went through a number of behavioral studies, and basically with fear conditioning, contextual fear conditioning, then we see this deficit. We don't see any deficit in the SLC6A1 heterozygous mice, but we do see it in the homozygous knockout mice, and we're able to recover some of that ability upon treatment. And kind of similar thing, if we're looking at nest building, hind limb clasping, rotor rod, these are all phenotypes that we could quantify in the homozygous knockout mice. And we could rescue it with gene therapy when we treated the newborn mice with an ICV administration. The heterozygous mice don't really show a phenotype. We have the EEG phenotype, but as far as behavioral deficits, we always see sort of trends where they're going a little bit lower, and you see that in the rotor rod where these are the heterozygous mice. Well, sorry, on this we don't have the wild type mice on here, but they usually run a little bit lower. Actually, this black line. They would run a little bit lower than wild type mice, but it's not significant. In this case, when we treated the heterozygous mice with AV, they did improve a little. They trended to improve on the rotor rod phenotype, but again, these differences weren't significant. Okay, so that's all neonatal treatment, and and one of the sort of bad consequences of this was we did see when we treated these newborn mice with this treatment that we did get some adverse effects. Actually, about 18% of the mice died. This was very concerning to us. We also went from having something in the mice that looked more reminiscent of the human seizures, of more of like absent seizures, to some of them developing these convulsive seizures. And so there was, again, when we're treating newborn mice with these high doses of this vector, we were seeing adverse effects along with all the therapeutic effects. And this was much worse if we used a non-specific promoter, something that wasn't neuron-specific, and all that's in the paper. Okay, so the question arose of, can we rescue the EEG phenotypes at older, more translationally relevant ages? And if you're treating newborn mice, that's developmentally equivalent to treating in utero in a human, like before birth. So we do the sort of the newborn mouse studies as a proof of concept, but it's not something that we feel would really support a human translation. And so we started off doing intrathecal injections at 5 days old in these mice, and that would be more reminiscent of a human infant. And then doing behavioral tests and EEG recordings. And so, let's see. So basically, we have the heterozygous mice here with an abnormal EEG tracing, much worse in the homozygous knockout. And then we saw, really a normalization of that EEG pattern when we treated the heterozygous mice, but not when we treated these severe knockout, the homozygous knockout mice. And this is just quantified here. where we were trying to establish a dose response, but at these doses there really wasn't a dose response, where we reduced the seizures significantly in the heterozygous mice, but not in these severe homozygous knockouts. And one of the things that pointed us to, and I kind of discussed this when I presented it last year, we discussed this extensively in the paper, is just, I think doing a lot of experiments in the homozygous knockout mice, those are much more severe than the patients, and I think that they're probably much, much harder to rescue. And I would argue that they're not necessarily relevant to be trying to treat them. Unfortunately, that leaves us with a heterozygous knockout mice, which have this EEG phenotype, which is significant to the patient population, but we're not able to really evaluate therapeutics in terms of treating the behavioral aspects of this disease. So humans, I'd say, are kind of like in between these two mouse models. So this is the newer data that's not in the paper, which is one graph, but it was a lot of work, where we were basically trying to establish a dose response and also an age response to treatment. So we were doing the same intrathecal injections of this vector in the heterozygous mice, and then just measuring EEG at 4 months post-injection. And so we treated them at 15 days old, 10 days old, or 5 days old, and we did a dose range. Honestly, I didn't expect this to work at 15 days old, but we did get a reduction in the seizures that was significant. And we kind of saw across the board there was this partial reduction in the EEG phenotype, and it was across all ages, across all doses. So this was actually encouraging, and I think the next steps to this is we want to take it later than P15 to see if we can extend this out and have a benefit treating later. And we also want to go into lower doses here, lower than what we tested. And I'll say the lower doses, like at P10, 1.4 e to the 11, that we tested at P5 and P10, that's actually lower than the doses that we're using in our current AAV9 clinical trials. If you extrapolate that to humans, it's about 60% of the dose that we're dosing across multiple clinical trials. So this is entirely feasible to translate those doses into humans, as long as all the safety data continues to look good. The other thing is in the paper that we published, we did pretty extensive safety studies in mice at older ages, at like 7 days or older, and we didn't see any safety consequences at all. There were no adverse effects if we treated mice at 5 days old or older. All the safety concerns that we saw were really limited to dosing those newborn mice. So this is the summary here. The neonatal ICV injection, I'd say, offers encouraging efficacy in both models, the heterozygous and the homozygous knockout models. But in our view, treating this neonatal age at the dose that we did, which was fairly high, had unacceptable adverse effects. Treatment at 5 days old and older in the homozygous knockout mice provided minimal or no benefit. But when we treated the heterozygous knockout mice, which genotypically matched the patients, we saw, I'd say encouraging efficacy treating them at 5 days old, 10 days old, or 15 days old. And this is really around rescuing the EEG phenotype. So, and as I said, dosing all the genotypes, including wild-type mice at 5 days old or older, was well tolerated. And we took, a lot of these mice out to a year old. And this is just kind of the summary of our efficacy data. And like I said, there's a lot more information in the preprint that's available. Let's see. I think I covered this pretty well. I'd say we still haven't determined a minimally effective dose for human translation. This is something that the FDA would want to see. We basically need to titrate the dose down to a point where it doesn't work. We need to determine if the treatment can be efficacious after 15 days old in mice. And I kind of made this point last year, and I'll make it again. I think that the homozygous knockout mice are probably not the best model to test treatments in. It might be good for a first pass if you're treating newborn mice, but otherwise, I'm not sure that they're going to be very amenable to any kind of rescue at older ages. So I want to acknowledge my team works really hard every day, especially Ray, who's been leading this project along with Matt here. Caddy Kang was a really key collaborator, especially early in this project, that helped us test some of our constructs in vitro models and providing some mouse models to us. And SLC6A1 and Amber in particular are the only reason I'm here and the only reason I started working on this. And then we did have funding from Taysha Gene Therapies. They were kind of a commercial partner for the last few years, but due to market conditions and whatnot, they've backed out now. So anyway, I appreciate everybody's attention. I don't know if we have time, but I could take a couple questions. Oh, yeah, Caddy.
You see convulsive seizures, right, after treatment. In your heterozygous, did you say that?
I think it was across, when we injected the newborn mice, it was mixed litters. So we had, I think, some wild type mice, some heterozygous mice, some knockout mice. We saw it across all genotypes. Yeah.
I see. Second question is, it's kind of odd. Did you see anything in the homozygous when you inject the P5? So I would expect you will see some improvement in the rotor rod.
Yeah, so when we injected the homozygous knockout at P5, we saw improved nesting behavior, but nothing else. Maybe there were a couple other behaviors that were trending a little bit, but it was really, the EEG was not affected at all. Nesting was the one thing that we were rescuing across even older ages. That was the easiest thing to rescue, is the nesting behavior.
Oh, it will work now. I have to say Steve 3 times for it to work. Great progress since last year, thank you. I have 2 questions. One, which endpoint will you use for determination of the MED? So what will be your parameter? And the second is, are you planning to get into the clinic with ICV or IT administration?
So in terms of the minimally effective dose, really our only readout that we can use is the EEG phenotype. So that's what we're using to drive our dosing, along with the planned GLP safety studies. And then what was the other question?
Route of administration.
The route of administration. Right now it would be a lumbar intrathecal injection with Trendelenburg positioning and whatnot. That's what we're doing for all of our other trials.
So you're not transitioning to ICV?
Not right now.
Okay, thanks.
You mentioned improvement at 5 days, 10 days old, and 15 days old. Was there a difference in the amount of improvement as the mice were older? Did you see less improvement than you did in the 5-day-old mice, or was it pretty consistent?
Yeah, so comparing like the reduction or the improvement in the EEG patterns at 5 days old, 10 days old, 15 days old, it was probably trending towards working better at younger ages, but really we didn't, there was no significant kind of age effect. So it was still working at 15 days old. I think it was more significant at 5 days old. But we only tested the high dose at 15 days old. So if we titrated the dose down, maybe we would start to see it tail off. That's what I would expect. I was really expecting to see a dose response and also an age response, and we weren't seeing that to a significant effect.
It'd be interesting to see
Yeah, yeah. Yeah, that's a really fuzzy issue. All of these, like up to 15 days old in mice, we're talking about sort of pre-adolescent children. All right, thank you, everyone. And I guess for all the, family and patient advocates in the room, I will have like a lay version of the talk tomorrow morning.